they're a hot topic because they're basically transforming the oncology space because they're a precise way to get the payload to the tumor, and they optimize the drug to antibody ratio. Thus, they maximize tumor destruction and they minimize damage to healthy cells.
Hello, you're watching Proactive. I'm joined by Dr. Kristen Buck, the Chief Medical Officer at Lisata Therapeutics. Kristen, very good to speak with you. You've announced a research agreement with Catalent to evaluate certepetide using Catalent's Smart AG antibody drug conjugates platform. Can you tell us more about this license?
Yes, so nice to be here today. Thanks for having me. Yeah, we're really excited to have a research license with Catalent. They're a leading CDMO. The research license is evaluating certepetide as a payload to evaluate certepetide preclinically with Catalent's SMART-TAG dual payload ADCs. Basically, Catalent will assume all R&D expenses. Lisata will provide consulting support and receive an upfront payment and potentially future considerations based upon the results of that preclinical evaluation.
Can you tell us more about certepetide and its tumor targeting and penetration technology?
Sure. Certepetide is actually a really unique asset. It's a cyclic 9-amino acid peptide that specifically binds to requisite receptors that are upregulated on most solid tumors. Once bound to these receptors, it gets cleaved into a linear peptide that activates a CN rule active transport mechanism, which actually enhances the penetration via an endocytic pathway of anything that's in the circulation at the time, most likely anti-cancer therapies. This increases the penetration of these therapies and leads to typically better outcomes in patients with solid tumors. Certepetide also modifies the tumor microenvironment, which is usually immunosuppressive, and enhances cytotoxic T cells and inhibits regulatory T cells, making the cloak of the cancer better able to be recognized by the patient. Further, certepetide works agnostic to the anti-cancer modality with which it's given, so it can work with current therapies and emerging therapies. And lastly, certepetide is unique in that it has a very benign safety profile, so it doesn't enhance or worsen the therapies with which it's given safety profile.
So maybe looking at ADCs, what do they do and why have they become such a hot topic at the moment?
Yeah, ADCs are antibody drug conjugates. Basically, they're an innovative biopharmaceutical product in which a monoclonal antibody is linked to a small molecule or drug, typically a cytotoxic agent or anti-cancer agent, with a stable linker. I like to think of them as a biological missile, which basically delivers chemotherapy typically to cancer cells. Minimizing damage to healthy tissues, right? So a monoclonal antibody is typically directed to a target or a tumor-associated antigen on the surface of a malignant cell. And what does that do? It ensures precise delivery to cancer. The linker on that antibody keeps it stable in circulation, enabling a payload attached to the linker to get directly to the tumor. And once it gets to the tumor, the payload gets released by cleavage, and therefore the cytotoxic drug gets right to the tumor. Why are they hot? They're a hot topic because they're basically transforming the oncology space because they're a precise way to get the payload to the tumor, and they optimize the drug to antibody ratio. Thus, they maximize tumor destruction and they minimize damage to healthy cells. And I think actually they're going to transform other therapeutic areas because they actually allow precision medicine to different types of antigens.
So what's the scientific rationale then and the potential of combining ADCs and certepetide?
So as I said, ADCs use monoclonal antibodies to get to tumor-specific antigens, and thus they ensure delivery of the payload, in this case cytotoxic drugs, to kill cancer cells. But there's a problem with cytotoxics actually getting into cancer cells. In the past, we used brunt force, basically giving more cytotoxic drugs to get into the tumor microenvironment because they're covered with a dense stroma, not allowing the cytotoxics to get in. But when you give certepetide, which enables tumor penetration, you ensure that the payload, when given with a payload of certepetide, actually penetrates deep into the tumor. And so we allow not only targeted delivery, but enhanced penetration and thus hopefully better efficacy. Further, the presence of certepetide in that tumor microenvironment reduces the immunosuppressive nature of the tumor microenvironment and reduces the chances of metastases, hopefully increasing patient response and better outcomes with the ADC therapy.
Kristen, what's your expectations for initial data later this year, and what positive data could actually do for the relationship with Catalent?
We really hope to receive results from this collaboration in the near future, hopefully maybe before the end of the second quarter. And our hope is that positive results could lead to further solidification of our collaboration with Catalent.
Kristen, I hope you'll keep us updated on any progress, but thank you very much for taking the time today.
Really, thank you for having me, as always.
It's Dr. Kristen Buck, the Chief Medical Officer at Lisata Therapeutics.